record this today. So last time, yeah, we talked about the Kootenay Lake in the Kootenays in British Columbia and the experiment of adding mysid shrimp. And that was a story, a true story, but it didn't just take place in BC, it took place in a lot of other countries. And there's been other experiments that have taken place without being fully aware of the repercussions of implementing certain strategies to try and increase productivity. And one thing I wanted to do today is describe productivity. I'm not sure if we defined it very well last time. So let me give you a definition of productivity. I'm just going to do it right here on this page. Productivity. So primary productivity will be the productivity of plants, the photosynthesizers. So productivity is the rate of, excuse me, productivity, the rate of new biomass accumulating at any level. So primary productivity is the productivity of the producers, the autotrophs. And I'm also going to send you, I watched it briefly today, a Khan Academy description of productivity, because it breaks it down very, very well. So let's look at productivity. So primary productivity is limited by the amount of energy input. As you can imagine, the amount of sun input. So there's more productivity in those places we looked at before, because, where was that? That was back here. We had some of our very productive areas, like the tropical rainforest, for example, because sun and water are not limiting. And nutrients probably aren't limiting there very often either. In some cases, even trace elements may limit productivity, primary productivity. So this is the Sargasso Sea, excuse me, which is off the Gulf of Mexico. And there have been studies done there about nutrients added to an experimental culture and the relative uptake of carbon-14 by those cultures. So carbon-14 is an isotope. So when organisms take it up, it can be detected. So carbon dioxide is taken out of the atmosphere by plants through photosynthesis, and a proportion of that carbon is carbon-14, which can be traced and detected. So relative uptake of the control is 1, so that's an arbitrary number given to uptake of a control, that whereby the nutrients have not been added. If nitrogen and phosphorus are added, then it goes up slightly. If phosphorus and nitrogen and plus other metals were added, it also went up slightly. If nitrogen, phosphorus, and metals, including iron, are added, the relative uptake went up considerably. And when nitrogen and phosphorus and iron were added, it also went up considerably. So the uptake of carbon-14 uptake measures primary productivity. It's a measurement. So that's what ecosystem ecologists do. They measure things like productivity. And this is one of the ways in which they do that. So this shows that in the Sargasso Sea, at any rate, iron is limiting primary productivity. The addition of large amounts of nutrients to lakes has a wide range of ecological impacts. This is a story of eutrophication. So eutrophication, adding nutrients to an aquatic ecosystem. That can be lakes or rivers. May lead to eutrophication. In other words, making a lake or a river more nutrient rich than it was before. So increasing nutrient richness. There are naturally eutrophic lakes. They just have a lot of nutrients from the surrounding vegetation. The other kind of a lake would be an oligotrophic lake. Oligotrophic means nutrient poor, and eutrophic means nutrient rich. And so I'm telling you this because nutrients are another addition that humans have added to a lot of aquatic ecosystems. So these are some lakes in Ontario, and a researcher named, what's his name? I think it was Richard. I could be wrong about that. I know his last name was Schindler. And he investigated what was happening in lakes in Ontario. So lakes in Ontario were experiencing algal blooms. Algal blooms. In other words, an enormous growth of algae, and not any kind of algae. This was cyanobacteria. In particular. And the cyanobacteria can cover a lake and cut out the sun for the other algae in the lake, thereby causing fish kills quite often. And that's what was happening in Ontario. Algal blooms, cyanobacteria, cutting out light, causing sometimes fish kills. So a large number of fish dying in the lake. So he questioned why that was. What was it that was causing the cyanobacteria to be successful over all the other bacteria, and what was being put into those lakes? So as it turns out, cyanobacteria is a nitrogen fixer. So you can add nitrogen and phosphorus to a lake, and the more phosphorus you add, the better for the cyanobacteria because it can make its own nitrogen. And it turns out that, so maybe I should write this down, nitrogen and phosphorus are important nutrients, not just in lake systems, but also in terrestrial systems. If phosphorus is added, cyanobacteria will bloom because for it, nitrogen is not limiting. Therefore, it grows uncontrollably and outcompetes the other algae. And you may be wondering why were there so many phosphates added? And that is from detergent. So it turns out that it was detergent that was being run off into lakes and streams that had all these phosphates in it. So now, because of Dr. Schindler and his research, detergent is largely phosphate free, and that's the reason. So that was really good research done to alleviate the effects of phosphates in detergents. I thought that was very interesting. So temperature and moisture affect primary production on a large geographic scale. How do we measure that? By evapotranspiration. We can measure how much water is transpired by plants. So what evapotranspiration is, it's the amount of water annually transpired by plants and evaporated from the landscape. And it's related to net primary production because the more a plant grows, the more it uses oxygen, and the more it uses carbon dioxide, and the more evapotranspiration there is. So in this case, we're looking at net primary production on the y-axis and evapotranspiration on the x-axis. So this just shows that there's a correlation between the measurement of evapotranspiration and the measurement of primary production. So as you would expect, tropical forests are very high on that scale. They have high productivity and high evapotranspiration. And temperate forests are around the middle, and tundra and shrubland are around the lower end. Nutrients in a local scale, as we saw with the lakes, some soil nutrients can be limiting. So this is a study of phosphorus in a salt marsh. So this shows biomass on the y-axis and time on the x-axis. So it's June, July, and August. The study was done quite a long time ago, 1980. So the control is down here in red. So nothing was added to the control. Phosphorus only was added here. It's not very different from the control. But when only nitrogen was added, then productivity went up. So it looks like the nitrogen was the limiting factor for growth. So one really important concept, I'll just admit this person. One important concept is that the efficiency of biomass transfer is quite low between trophic levels. So far we've been talking about primary productivity, but there is secondary productivity as well. And that's the amount of chemical energy in the consumer's food converted to its own new biomass in a given period of time. New biomass. It's not all converted to new biomass because individuals require some of that for cellular respiration. So I'll just write that down. I'll write that down. Energy transfer is inefficient because some of the lower level that is being consumed is indigestible. Like in plant cellulose, for example, we can't digest that. A lot of animals can't digest cellulose, so it just simply goes through the system and gets pooped out. It's indigestible and pooped. That is the technical word, pooped out. Also, some of that energy is used in cellular respiration. So instead of being stored as biomass, it's used up in cellular respiration just to maintain the metabolism of the individual. Maintaining I guess it's a basal metabolic rate, but we'll just say basic metabolism. So it doesn't get converted to new biomass. So when a caterpillar, for example, feeds on a plant leaf, only about 1/6 of the energy in the leaf is used for secondary production. So in this case, energy is being measured in joules. So of the 200 joules, 100 joules lost as feces, 67 joules in cellular respiration, and only 33 joules become new biomass. So net primary productivity is equal to gross primary productivity minus R. So glucose produced during photosynthesis is gross primary production. Some is used to drive cellular processes, respiration. Some is available for new biomass. It's only the new biomass that is the net primary production. So this is a simplified equation to show what net primary productivity is. So a fraction of the energy stored in food that is not used for respiration is converted to biomass in the next level of the trophic chain. It ranges between about 5 to 20%. It's good to look at pyramids because they show graphically the energy that is lost. And it shows it by portraying either the energy per level or the biomass per level or the individuals per level. In this case, we're looking at joules again. So this bottom bar portrays the 10,000 joules of energy of the primary producers. Only 1,000 joules of energy is available for new growth of the primary consumers. In this case, it looks like a locust or a cricket. And then that gets much smaller, 100 joules only for secondary consumers and only 10 joules for the tertiary consumer. So that's why trophic levels are only usually 4 to 5 levels. You don't find food chains that have 7 or 8 or 9 tiers. They just simply don't occur in nature because you need quite a lot in order to sustain any given level. You need a lot of input of energy. So that shows energy loss. This is a biomass pyramid. So not an energy pyramid, but a biomass pyramid. This is from a place called Silver Springs, Florida. This is where this data came from. Here's the primary producers at 809 grams per meter, per square meter. And you can see how much less there is of biomass as you go up the trophic levels. Interestingly, some aquatic ecosystems have this weird inverted biomass pyramid. So here, the primary producers have a smaller biomass than the primary consumers. So this is data that's come from the English Channel. A very small standing crop of primary producers. Why do you think that is? Can you offer an explanation why that might be? I don't expect you to know that, of course, but you could guess. It's not a simple, straightforward answer. What it is, as it turns out, is that these primary producers of phytoplankton have a really high turnover rate. So at any given moment, there's not a large biomass, but they turn over all the time. As the reproduction is so high, the reproduction rate, that there's a constant turnover and a constant resupplying of the phytoplankton to the zooplankton. So that's the reason why that happens. It's relatively rare, I think. This shows the number of organisms at each level. So this shows 5, almost 6 million, 708 or so thousand, less than half of that, and only 3 at the top. So if you're looking at number of individual organisms, it's a little bit problematic. So looking at number of organisms instead of biomass may show different pyramid structure. Because some individuals may be very large. So one individual may account for quite a lot of biomass, but it wouldn't be reflected in the numbers. How can I say that? Instead of biomass, they show different pyramid structure because individuals can be large or small. So how does this apply to the human population? So you and I. Eating meat, as it turns out, is relatively inefficient. So knowing what we know about energy transfer, you might be thinking, yeah, I get that. It took me a while to get this, but if you look at the trophic levels given beef, this is the primary producers here. I think this is a biomass chart. And here are the cows at this level, and this isn't a human. So the human in this case is a secondary consumer. So we're actually just showing in this graph which level humans may or may not be at. In this case, in this case, the human is a primary consumer. These are your vegetarians, which turns out is awesome for the planet. So what I'd like to show you now is a great video about why not eat insects. So that's the name of the video. And I want you to decide now whether or not you would eat insects as part of your diet. And then if your mind is changed by the end of the video or not. And this is a TED Talk. It's by Marcel Dickey, and he is Dutch. There's a nice Dutch accent, and we'll see what you think. There are 5 reasons, according to Marcel Dickey, that are good reasons for eating insects. Okay, so I'll stop there for now and we'll watch the video. Thank you.